For centuries, the House of Habsburg secured their dominance over Europe through a calculated strategy of royal intermarriage. They successfully consolidated crowns and territories, but this isolation exacted a steep biological toll, manifesting clearly in the severe misalignment of their lower jaws. By continuously reproducing within the same family line, the Habsburgs subjected themselves to intense consanguinity. They isolated their lineage from outside genetic input, progressively restricting their available gene pool with every generation. The units trapped within that shrinking pool are genes, specific sequences of DNA that code for heritable traits. Because the family interbred so frequently, certain alternative forms of those genes, called alleles, became highly concentrated. A person's exact combination of alleles forms their genotype, the hidden molecular code. The physical manifestation of that code in the real world is the phenotype. For the Habsburgs, their restricted genotype expressed itself as mandibular prognathism, the infamous Habsburg jaw. Inheritance operates as a rigid statistical mechanism. It demonstrates how microscopic molecular pairings, when constrained by human behavior, scale up to dictate the biological fate of an entire dynasty. All genetic variation, every new allele in a population, originates as a physical error during DNA replication. We call these errors mutations. The most common errors are point mutations, where a single nucleotide in the DNA sequence is swapped for another. Depending on the exact swap, the resulting protein might remain unchanged, substitute a single amino acid, or truncate prematurely with a stop codon. But when a nucleotide is inserted or deleted entirely, the result is a frame-shift mutation. Letter sequences called codons. Wedging a new nucleotide into the center shifts the entire downstream reading frame, radically altering every subsequent amino acid. A single misaligned base pair appears infinitesimally small, yet these microscopic disruptions generate the raw genetic diversity required for any species to adapt to a changing environment. To understand how these mutations pass from parent to offspring, we rely on the foundational mechanics identified by Gregor Mendel in the 1860s. Mendel's law of segregation states that an organism carries two alleles for every trait, which separate during reproduction. Mechanically, this occurs during anaphase 1 of meiosis, when homologous chromosomes physically pull apart so that each gamete receives only one allele. His second rule, the law of independent assortment, dictates that inheriting a specific allele for one trait does not influence the probability of inheriting an allele for an entirely different trait. The cellular engine driving this independence is recombination, or crossing over. During prophase 1, homologous chromosomes pair up and physically swap small segments of genetic material, creating novel allele combinations that did not exist in the parent. Here, meiosis functions as a biological randomizer. By continuously segregating and recombining chromosomes, the process guarantees that mutant alleles are shuffled into unique configurations for the next generation. Calculating the exact probability of an offspring inheriting these configurations requires a specific mathematical tool, the Punnett square. This grid calculates a dihybrid cross, tracking the inheritance of two completely dominant traits simultaneously. When crossing two individuals that are heterozygous for both traits, the mathematics dictate a strict 9:3:3:1 phenotypic ratio in their offspring. The primary exception to this mathematical rule occurs with linked genes. If two genes are located very close together on the exact same chromosome, they are rarely separated during recombination. This diagram tracks recombination frequency, denoted by the Greek letter theta. It calculates the statistical probability that linked genes will separate during crossing over, operating on the standard that one map unit corresponds exactly to a 1% chance of recombination. By identifying the physical distance between genes on a chromosome, we can calculate the precise statistical probability of those traits remaining linked and appearing together in future populations. Moving beyond a single Punnett square requires scaling our mathematics to encompass the millions of reproducing organisms that make up an entire species. To measure whether a population is evolving, biologists use the Hardy-Weinberg equilibrium. This acts as a biological null hypothesis, a mathematical baseline representing a gene pool that is completely stable and experiencing zero evolutionary change. Maintaining this requires 5 strict conditions. The population must be extremely large, Mating must be entirely random, there can be no mutations, no gene flow, and absolutely no natural selection. This graphic models a gene pool using the allele frequency equation, P plus Q equals 1. P is the dominant allele, Q is the recessive. To predict actual organism distribution, these frequencies combine into the genotype equation, P squared plus 2PQ plus Q squared equals 1. This maps the proportion of homozygous dominant, heterozygous, and homozygous recessive individuals. The Hartree-Weinberg equations provide a mathematical standard. The exact moment real-world population data fails to balance these equations, it proves that evolutionary forces are actively at work. When those equilibrium conditions break down and natural selection acts on genetic mutations, we use the modern synthesis model, sometimes called neo-Darwinism, to map the resulting changes to the gene pool. This curve illustrates stabilizing selection, a process that actively selects against extreme phenotypes. For example, human birth weights are maintained within a narrow, optimized window because fetuses that are significantly too small or too large face severe survival penalties. Conversely, extreme environmental pressure causes directional selection. When applications of the pesticide DDT killed off susceptible mosquitoes, the population's phenotypic norm shifted entirely, ensuring only the highly resistant insects survive to reproduce. Here, the normal distribution breaks entirely due to disruptive selection. Intermediate phenotypes are penalized, while both extreme variations are favored. On the Galapagos Islands, seeds were either very large or very small, strongly favoring finches with extreme beak sizes and eliminating the medium beak average. Continuous disruptive selection eventually splinters a single population. If the extreme groups remain separated for enough time, they achieve reproductive isolation, losing the ability to interbreed and finalizing the formation of new species. This phylogenetic tree maps 9 species of Saccharomyces yeast. Genetic analysis reveals that their evolutionary divergence closely correlates with their optimal growth temperatures, demonstrating how sustained environmental pressure forces a population to splinter into distinct species. Macro-level evolution is the mathematical consequence of differential reproduction. A single molecular mutation, having survived the statistical gauntlet of cellular division and environmental pressure, scales up to permanently rewrite the architecture of a species.